Hello all, welcome to my YouTube channel, that is Biochemistry Basics by Dr. Amit. Today's topic is electron transport chain. First definition of biological oxidation. Biological oxidation is basically transfer of electron from the reduced coenzymes through the respiratory chain to the oxygen is known as a biological oxidation, and the energy which is produced during this process is trapped as a ATP. So this coupling of oxidation with phosphorylation is called as a oxidative phosphorylation. And flow of electron occurs through successive dehydrogenase enzyme together known as a electron transport chain. And the electron flow from electronegative potential, that is minus 0.32, to electropositive potential, that is plus 0.82. So that is the definition of biological oxidation and definition of electron transport chain. And before going in the detail of electron transport chain, one should know that loss of electron is known as a oxidation and gain of the electron is known as a reduction. And the substance which exists in the both state, that is in the reduced as well as in the oxidized state, is known as a redox couple. So now let's see what are the various enzymes of biological oxidation or electron transport chain. So the basically enzymes of biological oxidation are belong to oxidoreductases, which includes first oxidases, second one is the dehydrogenases, third one is the peroxidases, and the fourth one is the oxygenases. Now let's see what this enzymes, what are the various function of this enzymes are. The first oxidases will carry out the removal of hydrogen from the substrate in the form of H2O or H2O2, and over here oxygen will act as an acceptor for the hydrogen, that is the oxidases. Second one is the dehydrogenases. So dehydrogenases basically it concerned with the removal of hydrogen from the substrate, but over here oxygen will not act as a hydrogen acceptor. For the hydrogen acceptor, there are the specific coenzymes are available in the dehydrogenases. So that is the main difference between oxidases and dehydrogenases. Third one is the peroxidases. So they act on the hydrogen peroxide. Hydrogen peroxide act as a substrate, and two particular types of hydroperoxidases are there. One is the catalases and another is the peroxidases. And another one is the oxygenases. Oxygenases are involved with the direct addition of oxygen into a substrate molecule. There are the two type of oxygenases. One is the monooxygenases and another is the dioxygenases. These are the information related to the enzymes of biological oxidation. Now, what are the various coenzymes involved in biological oxidation? The first one is the NAD+, that is nicotinamide adenine dinucleotide. Second one is the NADP+, that is nicotinamide adenine dinucleotide phosphate. Third one is the FMN, that is flavin mononucleotide, and the fourth one is the FAD, that is flavin adenine dinucleotide. So these are the four coenzymes involved in a biological oxidation. Now, location of electron transport chain. So the main location of the electron transport chain is they are located in the mitochondria, and inside the mitochondria, particularly to the inner mitochondrial membrane, inner side of the inner mitochondrial membrane. So that is the location of electron transport chain. So this you can see this is the inner membrane and this is the inner membranous space. So the location of electron transport chain, all the enzymes of the electron transport chain are located on the inner side of the inner mitochondrial membrane. This one is the matrix and these are the crista, and this one is the outer membrane. Fine, so that is the location of the electron transport chain. This can be asked in a various multiple choice question. Now, organization of electron transport chain. So mainly electron transport chain or respiratory chain, the electron are transferred from NADH to the chain of electron carriers. There are the various electron carriers are available. So the electron are transferred from NADH to this chain of electron carriers. And there are the four multi-protein complexes are there, that is called as a complex 1, complex 2, complex 3 and complex 4 in the electron transport chain. And all these complexes are connected by two mobile carriers. One is the coenzyme Q and another is the cytochrome C. So this all four complexes, complex 1, 2, 3 and 4 are connected by two mobile carriers, that is coenzyme Q and cytochrome C. So the name of the complex one is that is known as the NADH coenzyme Q oxidoreductase, and the transfer of electron occurs from NADH to the coenzyme Q. So that is the complex one. Complex 2 is known as a succinate coenzyme Q reductase, and the transfer of electron occurs from succinate to the coenzyme Q. That is the complex number 2. Complex 3 is the coenzyme Q to the cytochrome C oxidoreductase, and the transfer of electron occurs from coenzyme Q to the cytochrome C. And the complex 4 is the cytochrome C oxidase, and the transfer of electron or flow of electron occurs from cytochrome C to the molecular oxygen, which gets converted to the H2O. So this is the information related to the four main complexes of electron transport chain, which are connected to each other with the help of two mobile carrier. One is the coenzyme Q and another is the cytochrome C. Now let's see the complex one, that is NADH coenzyme Q oxidoreductase complex. So the first flow of electron or transfer of electron occurs from NADH to the FMN. So FMN gets converted to the FMNH2, and NADH H plus gets converted to the NAD plus. So there is a transfer of two electron and two proton from NADH to the FMN. At the end, FMN is getting converted to the FMNH2. Now this FMNH2 flow of electron from FMNH2 occurs to the iron sulfur protein complex, and this iron sulfur protein complex, after receiving the electron, gets converted to the reduced iron sulfur protein complex, and this iron sulfur protein complex transfer the electron to the mobile carrier, that is coenzyme Q. So this coenzyme Q gets converted to the QH2. Fine. So at the end of the the flow, we can say that flow of electron in the complex one is, and in addition to this, there is a transfer of 4H plus from matrix to the inner membranous space. In the complex one, there is a transfer of 4H plus from matrix to the inner membranous space. And we can say that flow of electron occurs from NADH to the FMN, FMN to the iron sulfur protein complex, and iron sulfur protein complex to the coenzyme Q. So this is the flow of electron in the complex one, which is known as a NADH coenzyme Q oxidoreductase complex. Fine. So that is a complex one. Now let's see complex 2, which is known as a succinate coenzyme Q reductase, and it is in a more it is this complex is in the close proximity with that of TCA cycle. So remember the TCA cycle step, that is the conversion of succinate to the fumarate, and there is a synthesis of one molecule of FADH2. Fine. Now this electron from FADH2 will pass to the again iron sulfur complex, and after receiving the electron, iron sulfur complex will be reduced, and this iron sulfur complex again will pass this electron, pair of electron, to the coenzyme Q. So coenzyme Q will get converted to the QH2. So, and over here in this complex 2, there is no pumping of any proton from matrix to the inner membranous space. So in the complex 2, there is no any kind of pumping of proton occurs. Fine. And we can say that flow of electron in the complex 2 is succinate to the FAD, FAD to the iron sulfur complex, and iron sulfur complex to the coenzyme Q. Coenzyme Q is the mobile carrier. Fine. That is about the complex 2. Now let's see complex 3. Complex 3, it is known as a coenzyme Q cytochrome C oxidoreductase complex, and flow of electron in the coenzyme in this complex 3 is there was a coenzyme Q accepted the electron from the complex 1 and complex 2, and it gets converted to the QH2. Now this electron from the coenzyme Q will pass to the cytochrome B, fine, and this cytochrome will be, after accepting the electron, will get reduced, and this cytochrome B will pass electron again to the iron sulfur complex. An iron sulfur complex, after receiving the electron, gets reduced, and this iron sulfur complex pass electron to the cytochrome C1, and this cytochrome C1, after receiving the electron, again it get reduced, and this cytochrome C1 will pass electron to the another mobile carrier, which is known as a cytochrome C. Fine. So that is the flow of electron from coenzyme Q to the cytochrome C. Fine. And we can say that flow of, and in addition to that, there is a pumping of 4H plus in the complex 3 from matrix to the inner membranous space. Mechanism for this pumping of 4H plus in complex 1 as well as in the complex 3 is not discovered yet. And we can say that flow of electron in the complex 3 is from coenzyme Q, fine, that is coenzyme Q, then FES, then cytochrome B, then the cytochrome C1, and then to the cytochrome C. Fine. Cytochromes are basically they are the ion containing hemoprotein which gets involved in the transfer of electron in the electron transport chain, and among all this cytochrome, that is cytochrome C, cytochrome B, cytochrome C1, fine. Cytochrome C1 is the water soluble and it is the mobile one, while all other cytochromes are the lipid soluble and they are fixed in the mitochondria. And in addition to take part in the electron transport chain, this cytochrome C is important for the apoptosis, which is also called as a programmed cell death. Fine. So that is complex 3. Now let's see the complex number 4. So complex number 4, which is known as a cytochrome C oxidase, flow of electron from cytochrome C, the oxygen, electron will transfer to the cytochrome AA3, and this cytochrome AA3, addition to the ion, it also contains copper ions. Fine, there is a presence of iron as well as the copper ion. So this cytochrome AA3, after receiving the electron, it will convert it to the reduced one, and this cytochrome AA3 will transfer electron to the molecular oxygen, and this molecular oxygen will get converted to the H2O. And in this complex, that is complex number 4, there is a pumping of 2H plus from the matrix to the inner membranous space. So that is all about the complex 1, complex 2, complex 3, complex 4. Fine. So flow of electron is cytochrome C, cytochrome AA3, and molecular oxygen. Now the main complex, which is concerned with the synthesis of ATP, that is called as a ATP synthase complex. And this ATP synthase complex is in main concern with the chemiosmotic theory. Find this chemiosmotic theory was given by the Peter Mischel. Fine. This theory states that synthesis of ATP in the complex number 5, which is also called as ATP synthase complex, is more related to the because there is a formation of protomotive force, because there is a pumping of H plus ion from matrix to the inner membranous space. So there is a more protons are available on the inner membranous space, so that leads to the creation of electrochemical potential difference, which leads to synthesis of protomotive force, and this protomotive force is responsible for the synthesis of ATP, which is called as a chemiosmotic theory. So coupling of oxidation with the phosphorylation. So that is the main chemiosmotic theory which was given by the scientist Peter Mischel. So let's see how this complex number 5 works. Basically, this complex 5 or ATP synthase complex is basically multi-unit transmembrane protein complex. It is composed of two portions. One is called as FO. It is not F0. O stands for oligomycin, because this FO unit is inhibited by the antibiotic oligomycin, and another one is the F1. This F0 and F1 are connected to each other, and they are the transmembrane one. FO is composed of the 4 polypeptide chain, while the F1 is composed of the 9 polypeptide chain, that is 3 alpha, 3 beta, gamma, delta, and epsilon. Fine. And this F1 projects into the matrix while this FO is located on the membranous side. Fine. So that is related to the FO and F1. And this FO act as a channel for the entering of proton. Fine. All the proton which is present on the inner membranous space will enter from this FO channel, and this F1 channel is concerned with the synthesis of ATP. Right? Alpha channel, alpha polypeptide chains are concerned with the binding site for the ATP and ADP, while the beta chains are concerned with the catalytical site for the synthesis of ATP. Fine, so that is information related to the FO and F1. And so this is you can see there is a high H plus ion which is present, fine, which is present in the inner membranous space. So this H plus ion through the FO unit will get entry into the through the FO unit will get entry, fine, into the matrix, fine, and there is a synthesis of ATP in the F1 unit through the binding change mechanism. Fine, as I have already explained that there is a binding site is available on the alpha polypeptide chain which is concerned with the binding site for the ATP and ADP, while the beta is pro polypeptide chain is concerned with the catalytical sites. Fine, so there is a binding change mechanism occur in the F1 unit, and that leads to the synthesis of ATP. Let's see the inhibitors of electron transport chain. This particular topic, that is inhibitors of electron transport chain, is very important for the various entrance examination. There are so many questions asked in a previous entrance examination like NEET, PG, AIMS. So please understand this topic very carefully. The inhibitors of electron transport chain. There are the three type of inhibitors of electron transport chain are available. First one is the inhibitors of electron transport chain proper. There are the site specific inhibitors are available. Second one is the inhibitors of oxidative phosphorylation that is concerned with the FO, F1 ATPase. That is the complex number 5th ATP synthase complex. The third one is the uncouplers of oxidative phosphorylation. These uncouplers are also important. And so let's see what are the various these inhibitors are. And so this is the diagram which shows the four complexes: complex 1, complex 2, complex 3, and complex 4, along with their mobile carriers, that is coenzyme Q, which connects the complex 1 and 2 to the complex 3, and cytochrome C, which is the connector between complex 3 and complex 4. The site specific inhibitors of electron transport chain basically are, so complex 1 and coenzyme Q gets inhibited by one antibiotic which is known as a pyrazidine A. Then it is also inhibited by phenobarbital and amobarbital, which are the barbiturate drugs, and it gets inhibited by rotenone also. So that is the complex 1 site specific. Then complex 2 is inhibited by malonate. Fine, the complex 2 is the succinate gets converted to the fumarate. Fine, and there is a synthesis of FADH2. So this particular is inhibited by malonate, and it is the example of competitive enzyme inhibition. And then complex 3 gets inhibited particularly by the British anti-leviside, BAL, and one antibiotic which is called as antimycin A. Then complex 4, which one is the important, that is the cytochrome oxidase complex. It gets inhibited by hydrogen sulfide, carbon monoxide, and cyanide. Cyanide poisoning affect the complex 4, that is cytochrome oxidase. And so these are the site specific inhibitor. Then inhibitor of oxidative phosphorylations are basically one which inhibits the complex 5. So the first one is the oligobenzene, and another is the ectrolyside, sorry, etractyloside. It inhibits the it is the inhibitor of oxidative phosphorylation. It basically inhibits the pumping of ATP outside the mitochondria and pumping of ADP inside the mitochondria. So that is etractyloside. So that is the inhibitor of oxidative phosphorylation. Now let's see the uncouplers of oxidative phosphorylation in detail. So this uncouplers of oxidative phosphorylation, what they will do, they are the chemical substances, they allow electron transport chain to proceed, but they prevent the phosphorylation of ADP to the ATP. So they will allow the electron transport chain to occur properly, but there is no synthesis of ATP. And so that is the and this substances are rather toxic substances, fine, and they are the lipophilic lipid soluble substances, so they can readily diffuse through the mitochondrial membrane, and they are capable of binding to the H plus ion. Now they can bind to the H plus ion. So because of their binding to the H plus ion, there will be no any protomotive force. There will be no any protomotive force will be synthesized because uh, there will be no any formation of proton gradient. Fine, that is the first thing. Now there is a no formation. So what will happen? There will be uh, the heat energy which is produced in the as there is no any protomotive forces synthesized, so the energy produced by transport of electron is released as a heat, fine, which is rather than being used for the synthesis of ATP. So these are the example of this is the mechanism of action of uncouplers of oxidative phosphorylation. Now there are the two type of uncouplers. A few are the example of various drugs and another are the physiological uncouplers. So let's see what are the examples. So the first example is 2,4-dinitrophenol, which was previously used as a drug for the weight reduction, and it was used in USA previously, but now it is completely banned now. Another is the dicomerol, which is an anticoagulant, and the third one is the salicyclate, which is a metabolite of aspirin. So these are the example of uncouplers, and another uncouplers are physiological uncouplers. So the first one is the thermogenin, fine, which is present in the brown adipose tissue, which is responsible for the thermogenesis in the newborn infants. Fine, and another is the thyroxine, and the another example of physiological uncouplers are bilirubin and free fatty acids. Thermogenin, thyroxine, bilirubin, and free fatty acid are example of physiological uncouplers. So this is all about the inhibitors of electron transport chain. Now let's see various multiple choice questions. The first question is, which of the following is a physiological uncoupler, which was asked in a NEET 2016, and the options are thyroxine, insulin, glucagon, and norepinephrine. And the answer is thyroxine. We all know that thyroxine, thermogenin, then free fatty acids and free fatty acid, they all are example of physiological uncoupler. Now let's see question. So about effect of 2,4-dinitrophenol. So 2,4-dinitrophenol is an example of, again it is example of uncoupler of oxidative phosphorylation. So effect is option A, O2 consumption increased, B, ATP is produced, C, respiration is decreased, and D, electron transfer is decreased. So the uncouplers of oxidative phosphorylation, what they will do, they will allow electron transport into occur, fine, but there will be no synthesis of ATP. So the answer will be the A, O2 consumption is increased. So answer is A. Multiple choice question is mechanism of cyanide poisoning, which was again asked in a NEET 2016. Inhibition of cytochrome oxidase, inhibition of carbonic anhydrase, inhibition of cytochrome C, and inhibition of ATP synthase. Along with cyanide, carbon monoxide, and hydrogen sulfide, they all inhibit the complex number 4, that is cytochrome oxidase complex. Fine. So inhibition of cytochrome oxidase. Then final acceptor of electron in electron transport chain is, which was asked in AIMS May 2014. Options are cytochrome C, oxygen, FADH2, and coenzyme Q. So the final acceptor of electron in electron transport chain is oxygen. Fine. After accepting the electron, it gets converted to the H2O. So that is B. Then dinitrophenol inhibits the electron transport chain by A, it inhibits the cytochrome B inhibits the ATP synthesis and electron transport chain, C inhibits the ATP synthesis but not electron transport chain, and D inhibits the electron transport chain but not ATP synthesis. So the answer is C, it inhibits the ATP synthesis but not electron transport chain, because it is an example of inhibitors, uncouplers of oxidative phosphorylation. So they allow electron transport chain to occur, but there is no synthesis of ATP because the energy being trapped is released as a heat. So that is answer. These are my references. Hope you have liked my video. Please like, share and subscribe Biochemistry Basics by Dr. Amit. Thank you.